Thank you very much. I'm Jonathan. I'm going to be talking to you today about Nanomix. This is just a brief outline of what I'm going to be discussing today. So the motivation for my work here is cell-free DNA analysis with nanopore sequencing. You probably have heard about cell-free DNA analysis earlier in the auditorium. Basically, cfDNA is accumulated in the blood by cells undergoing apoptosis. Whilst the majority of cfDNA comes from blood cells, tumors have been found to contribute cfDNA very early in tumorigenesis. Our goal here is to distinguish between the ctDNA and the cfDNA. Since normal cfDNA comes from blood cells, we can use methylation deconvolution to determine the cell of origin of the cell-free DNA. And if the cell-free DNA is coming from epithelial tissue, that's an indicator of disease. Methods. So we built a pipeline for end-to-end analysis of cell-free DNA using our tool Nanomix. After sequencing, we call modified bases using Dorado with the Remora model. We process the BAM file with our software MB tools, which creates a methylome TSV. We then call Nanomix to deconvolute to take that methylome and determine the proportion of cell types in the DNA mixture. The majority of the work here is on the deconvolution step, so I'm going to be going through a couple methods we use to deconvolute. Typically, methylation deconvolution is done using non-negative least squares approximation. Here, one chooses M loci that are differentially methylated in the constituent cell types. We measure the methylation level at each locus to get the sample methylome. On the other side, we have the methylation reference atlas, which consists of methylomes of purified cell types, the columns in the diagram here. Therefore, each element of the sample methylome should be some linear combination of a row in the atlas. And obtaining an optimum linear combination is going to be our objective. So we set the mixture proportion vector sigma to a free variable and optimize with quadratic programming. So previous work has shown this method to work quite well for methylation arrays, but what we found with our Nanopore data is that it didn't fit properly. And we hypothesized that this was because there was no attention to the signal-to-noise ratio. Basically, if you're sequencing at high depth in one region, it's going to be treated the same as sequencing at low depth in another region. So what we did is we developed a new deconvolution model called lllse. That stands for log likelihood with sequencing errors. And this is motivated by previous work called PERT, which is for expression deconvolution. Here, instead of defining our methylome with a single vector, we measure the probability of drawing M out of T methylated calls at each locus. We parameterize the binomial distribution with our atlas data and unknown sigma vector. We then optimize the sigma vector by maximizing with respect to the log likelihood. And the advantage of using a likelihood model is that it's more flexible to introduce parameters, particularly error parameters. When we started our research, the error rate for modifications on Nanopore was particularly high, and this was an item of concern. So we defined two types of error rates, P01, the overcalling rate, and P11, the correct calling rate. So if we let Z hat be the estimate of true methylation frequency, we can incorporate these error rates into our model and then define X hat like so. The only limitation here is that if you misspecify the error rates, the model doesn't perform that well. We also designed a second model, which we call MMSE, mixture model with sequencing errors. It also uses a binomial distribution, except this time we're going to be counting the number of methylated calls on each read and then assigning reads to cell types. Gamma here denotes the responsivity of read I belonging to cell type K, which is our expectation step. We then recompute the mixture proportion by taking the proportion of reads assigned to cell type K. That's our maximization step. And we use this expectation maximization optimization procedure to iteratively repeat until our likelihood converges. This model has an equivalent likelihood objective to LLSE, but it has the advantage of being able to assign reads to cell types. So I'm going to be going through a little bit of an overview of results on simulated data and real data. The advantage of using the likelihood model is that we're able to simulate methylomes of complex cell type mixtures from data in the reference atlas. In this experiment, we want to evaluate how models I described previously respond to increasing error rates and decreasing coverage. Here I'm simulating a methylome of 70% monocytes and 30% lung. The vertical facets in the plot show increasing coverage, and the horizontal facets are the models. You can see I added one more model here, lllsp. That stands for log likelihood with sequencing perfect. This is essentially the log likelihood model without the error parameters introduced. Within each facet, the overcall rate is on the x-axis and the correct call rate is on the y-axis, and we see that the MMSE model and the LSC model don't decrease in accuracy with increasing error, whilst NNLS and LSP increase in error with as a function of the simulated errors. We also observe that this relationship continues to even hold at 0.1x coverage, which is evidence that sequencing error and depth are critical to NNLS deconvolution. So while using simulated data is good, we wanted to evaluate these models using ground truths. So we created 3 benchmark data sets. And to create our ground truths, what we did is we sequenced purified cell types, and then we computationally mixed them at known proportions. The first 2 panels here show where we evaluate our models by mixing together lung cells and monocytes. We found that the performance was similar across all models, in contrast with the simulated data. Furthermore, we wanted to create a data set that models cfDNA. So what we did is we took a cancer cell line, HCT116, and PBMCs, and digested them with MNAs. MNAs is essentially an enzyme that's going to digest DNA unbounded to nucleosomes. So the coverage distribution is similar to what you expect from a cell undergoing apoptosis, like in cell-free DNA. Histogram here shows a regular pattern of nucleosomes every 150 base pairs. And what we found is that when we deconvoluted these samples using HCT116 as a purified cell type, we could mimic a residual disease monitoring application. And what happened was actually none of the models produced any significant results for deconvolution, so we wanted to investigate this a little bit further. So what we see is that the high error rate in the MNAs data is very unusual. What we did to measure the error rate is we compared the MNAs sample to a high coverage sample using R10 data, and we found that the error rate was around 0.07, which is unusually high. And there's a couple of reasons why short read data could be have a high error rate. In particular, what we looked at is there's an end repair bias. We observed that near the start of the read, the first 25 base pairs have a particular drop in the correct call rate. And what we hypothesize is that there's an end repair step in library preparation that does not reincorporate modified nucleotides. Additionally, we saw that there's a bias for heterochromatin in MNAs. So when MNAs is digesting around the DNA not bounded to nucleosomes, you're going to get an overpopulation of heterochromatin. And that's basically going to be associated with hypermethylated DNA. And since our deconvolution relies on the distribution of hypermethylation and hypomethylation, this is going to affect our results. So in conclusions, I do believe that Nanopore has the potential to be widely adopted for clinical use in liquid biopsy and cell-free DNA analysis because of its portable nature. And what we found is that methylation deconvolution of cell-free DNA here can be used for early cancer screening and residual disease monitoring, and sequencing error and depth are critical factors to accurate deconvolution. We see that due to the improvement of accuracy in the R10.4 chemistry and new base callers, that error modeling is not actually always necessary. And the critical factors in deconvolution we found with mock cfDNA is that there's a bias towards hypermethylation and errors towards the end of the reads. All in all, we built a pipeline for deconvolution, a tool, and benchmark data sets for further exploration of these issues. I'd like to thank everyone in my lab, our collaborators in the Young Lab, funding provided by the University of Toronto, and of course ONT for always providing us with flow cells. Thank you very much.